everyone. My name is Inga Rødahl and I am a field bioinformatics scientist at Pixelgen. And today I will walk you through our Proxiome Experiment Summary, which is an output you get after running the Pixelator pipeline. So running your FastQ files with Pixelator pipeline will give you a lot of different outputs. Importantly, the pixel files you can use for downstream analysis, but also this experiment summary in a HTML file. So when you open up your experiment summary, it will look like this. And on the first page, you have an overview of the different samples you've been running and the sample names as well as any conditions. And this you can also download as a CSV or an Excel file if you need it for any additional purposes. In this example, we have two PBMC samples, one that's unstimulated and one that has been treated with PHA to activate immune cells. The second tab is the quality metrics, and here we have a list of different metrics. If you're unsure what any of these means, we have this information symbol that you can hover above to get additional information about each of these metrics. The first thing we'll look at is the number of cells. And here we can see in sample 1 that we have about 1200 cells, and the second sample has 600 cells, so half the number. We then want to see that these samples have been assigned the correct amount of reads, and we can look at the total reads down here, and we see that indeed the first sample has received about twice as many reads as the second sample, which would be appropriate according to the cell numbers. Another metric to see if they have been sequenced substantially is to look at the saturation. Here we have the graph node saturation, which represents the proteins, and then we have the graph edge saturation, which is the link is in between each protein. We want the graph node saturation to be above 70%, which we see it's well above for sample 1, whereas for sample 2 it's almost or basically at just the threshold. Then for the graph edge saturation, we want it to be above 40%, and that is the case for both of these samples. We can also look at the saturation in these sequencing saturation plots. And here we have both the samples, which you can highlight separately. And what you want to see is that they kind of start to plateauing at the end of the curve. Next, we can also look at the number of reads that have been put into each step of the Pixelator pipeline, but also the reads that have been filtered away. So in this bar plot, the red color are reads that have been filtered away. And again, you can look per sample. And you also get this information in a table where you can see the frequency of reads that have been filtered. Again, also available for downloading. We then also want to look at the amount of reads per cell. So what we have here is we want to see a minimum of at least 250,000 reads per cell. And based on having twice as many cells, but also twice as many reads, as expected, these two samples then have fairly similar amount of reads, just above the threshold that we set. The number of cells is determined by the Pixelator pipeline, and one of the threshold is a minimum number of molecules that we want to see per cell. And we can visualize by ranking the cells by the number of molecules. Cells and components are the same thing in this case. And what you want to see in this molecular rank plot is that each of your sample gets this Ni structure in the plot. And the minimum threshold should then be below this Ni. The Pixelator pipeline will automatically find the best minimum threshold, but it is automatically set to a minimum of 8,000, which is the case for both of these samples. In your downstream analysis, you can also determine to change this threshold to suit your sample the best. We do, however, have a minimum median number of molecules per cell, which is at least 20,000. We see that both of our samples have well above that number. And also in this plot we see that the second sample, which is the stimulated cells, have a lot higher number of proteins per cell compared to the unstimulated. And this would make sense if the immune cells are activated and they start to upregulate a variety of different markers. However, what this means is that even though these samples have a similar amount of reads per cell, it means that for sample 1, these reads are split across fewer molecules than in the second sample. And this is something that we see as a result in the corners of our graphs. So each cell is represented by a graph, and we want the nodes, which represent the proteins, to be well connected in this graph to ensure that we are looking at good cells. And what we see is that the first sample has a really good connectivity with a corners of 2.4, whereas the second sample is just above our threshold, which is 1.5, and here at 1.7, indicating that these cells probably could have done with a little bit more reads to get a better k-corners of the graphs. So this is an example where you would have to take the nature of your sample into account when you plan your sequencing. Now the last quality metric is our control markers. So in our panel we have 3 isotype controls, and this is showing the amount of reads that have been allocated to these markers. And both of our samples seem to be at good quality with well below 0.1%, indicating that the cells have a good quality. So overall, from these quality metrics, we now know that our cells have good quality and they have been sequenced just enough. And the next tab in the experiment summary will then allow you to look at some automated cell annotation. So the experiment summary will cluster your cells and do a UMAP visualization. Here you see the clusters, and then here the conditions are colored with the unstimulated in blue and the PHA stimulated ones in brown. You can also look at the clusters split by sample, and you can see that this is quite a change between the two different conditions. Then the experiment summary also does a automated cell annotation based on a reference PBMC dataset. We do suggest to take a bit of caution in interpreting this annotation, especially if you're running any cells that are very different from PBMC. So, for example, a cell line or a enriched population. So in those cases, this annotation may be incorrect. But in this case, we are running PBMCs, so we can have a look at the output. And first you get an overview of the fraction of the cell types per sample. So in this case, we see that in the stimulated sample, there is an increase of, for example, CD8 T cells. Then you can also look at the numbers in a different manner, here split by sample, and you also get the actual cell number. And you can split between samples, and then you can see how the fraction is changing between. Now we can look at the UMAP visualization of this, and here you see the different cell types as described with CD8, CD4, NK cells, monocytes, B cells, dendritic cells, and also platelets. The experiment summary also does a second level of annotation, which is a bit deeper, where it, for example, annotates naive CD8 T cells from memory cells. Then you can also look at what these clusters and cell types are expressing based on the abundance. And here you find each of the clusters and with the cell type annotation. And for example, you can see that the T cells are indeed expressing CD3. And in this list, you can find your favorite markers to annotate and to double check the annotation. We can also look more into the abundance in this section of the experiment summary. And here you get the list of all the 155 markers. And you can look at this globally just between the two samples in this case. And we know that these have been resting and stimulated, so then we can look at an activation marker, which we can see is indeed higher on the stimulated sample compared to the resting one. You can also look per cell type, and we can then confirm the annotation again with, for example, CD3, which is expressed on the T cells. In these plots, it's only some major cell populations that have been shown. So if you're missing your favorite cell type in this experiment summary, you can always look at it in the downstream analysis. But this can at least give you a nice indication and confirm any potential annotation. So another marker that we can look at is, for example, CD8, which is expressed by the CD8 T cells, and we can also look at CD19, which is expressed by B cells. And again, this is split by sample, so if we look at another activation marker, we see that this is increased in abundance in all the different cell populations shown here. Next, we can also look at the spatial metrics, and this comes in a log2 ratio, which is our proximity score, and this is a comparison of the log2 transformed ratio of observed counts to expected counts of these linkers between different proteins. This is described in detail here, and you can also look further at our documentation. So this comes into different tabs. Initially, we have the clustering, which is how one protein is in proximity to itself. So here you can look globally at any marker that you prefer. We can look at ICAM1, for example, which you see here is slightly up in the second samples globally. And then when we look at per cell type, we see that ICAM1 is actually clustering on multiple different cell subsets. And ICAM1 is important for activation of immune cells to facilitate cell-to-cell interaction. We can also look at the colocalization, which includes the scores between different proteins, but it does also include the clustering here on the diagonal. I will zoom out a bit so we can see the whole heat map. So these heat maps only include the top 40 markers based on the proximity scores, and each marker has to be present at least 25% in the sample. So here again we can look at globally, and by visual inspection we can see that quite a bit of changes are happening in the proxiome from sample 1 to sample 2, which is the activated ones. But we're maybe more interested in looking per cell type. And again, by visual inspection, you can compare between the different samples that there are quite a few changes happening for each cell subset. So now I'm just going through each of them. And then lastly, also the B cells. So here, again, you can see there's quite a spatial reorganization happening. For example, for the B cells, you see that we have these different networks of proteins that seem to be changing when you go between different conditions. And this includes this network with CD19, CD21, CD72, which are all markers that are important for B cell function. So this allows you to kind of get an initial overview of what might be happening spatially on your cells. And then in the downstream analysis, you will be able to more specifically look at potential differential spatial metrics as well. Now the last tab in the experiment summary is just the run settings for the experiment summary, if you're interested by that. Now, I want to end on saying that if there is any issues, if you have any questions regarding the experiment summary, maybe if some of the quality metrics are looking a bit funky, please do not hesitate to reach out. You can find us on our website under data analysis support, and if you go here to contact data analysis support, you will get this form that you can fill in with any questions you might have. You can also reach us at support@pixelgen.com. Thank you very much for your attention, and best of luck with your experiments.